Hey guys, I'm Finn, and today we're diving into the exciting world of gene therapy, where science meets a bit of magic. Gene therapy, also known as gene editing therapy, is a cutting-edge approach that aims to treat or even cure diseases by modifying a person's genes. We'll explore two superstar techniques, CRISPR and viral vectors. Imagine if we could fix genetic diseases at their source, like swapping out a broken part in a car. That's exactly what gene therapy does for conditions like cystic fibrosis or sickle cell anemia. Today we'll focus on two methods, CRISPR-Cas9, also known as CRISPR, which acts as molecular scissors, and viral vectors, which serve as gene delivery mail carriers. Let's kick things off with viral vectors. Think of them as friendly viruses that have been reprogrammed for good. First, scientists remove the harmful parts of the virus so it can't cause disease. Then they insert a healthy copy of the target gene into the virus, essentially turning it into a delivery system. Finally, the virus delivers this package into the patient's cells, helping them produce the right proteins. A great example of this in action is Luxturna, a therapy that uses viral vectors to treat a rare form of inherited blindness. Viral vectors are particularly effective for treating diseases caused by a single faulty gene. They work especially well in areas like the eyes or brain, where cells don't divide too quickly. However, there are a few challenges. Sometimes the body's immune system can react to the virus, and once the gene is delivered, fine-tuning its activity can be tricky. Now let's talk about CRISPR, a tool that's as precise as a master sculptor's chisel. Here's how it works. A guide RNA first locates the exact spot in the DNA that needs fixing. Then the Cas9 enzyme acts like molecular scissors, cutting the DNA at the targeted location. Once the cut is made, the cell repairs the break, either by disabling the faulty gene or swapping it out for a healthy one. A recent success story from 2023 highlights how CRISPR was used to edit genes in bone marrow cells, allowing patients with sickle cell disease to produce healthy hemoglobin. CRISPR is incredibly precise, often making permanent changes that require fewer repeat treatments. However, there are some risks. One concern is the possibility of off-target effects where CRISPR might make unintended cuts. Additionally, certain tissues, like those in the brain, are harder to reach with this method. So how do these two techniques stack up? Viral vectors work by delivering a brand new gene, while CRISPR edits the existing gene. When it comes to precision, viral vectors are effective, but not as pinpoint accurate as CRISPR, which is laser focused on its target. In terms of longevity, viral vectors may require follow-up treatments, whereas CRISPR often provides a one-and-done solution. Each method has its risks. Viral vectors can trigger immune reactions, while CRISPR carries the risk of off-targeted cuts. Ultimately, both techniques have unique strengths, and the best choice depends on the condition being treated and the targeted tissue. In summary, gene therapy is paving the way for a future where we can truly fix genetic problems at their source. Viral vectors act like trusty delivery trucks, while CRISPR serves as the precise tool for permanent repairs. Who knows, one day they might even team up for an even more powerful treatment. And if you'd like to find out more about these techniques, check out NIH's website,